Today we're going to look at protein synthesis. This is the part one of two parts of this particular PowerPoint. I broke it into so it will be a little bit easier for you to digest. The two halves do go together from an original PowerPoint, so please do make sure that you watch both of them. We're going to start with a little introduction or a review of what the central dogma is. And then we're going to go right into the first part of protein synthesis, which is transcription. So the central dogma of biology is illustrated by the diagram that you're seeing here now. It can be broken into four major stages. So the whole idea is basically the journey that DNA needs to go through in order to support living organisms. So top on the diagram here is replication, and we've already talked about replication in class, so that's not a new concept for you. We understand that that's what happens when DNA is preparing the cell for cell division. So we have to duplicate our DNA, and that's the process of replication. So other than when we're dividing, replication does not occur, but the DNA is a very busy place. We need to have a way of accessing the information that's in the DNA and using it to carry out all of our life activities. So the rest of the central dogma is about how we are able to use DNA to take instructions to do the things that the cell needs to do. DNA, as we understand, holds a code, and DNA is unable to leave an intact nucleus of a eukaryotic cell. So the nuclear envelope, the double envelope that defines the nucleus in a eukaryote, is impenetrable to DNA. The DNA code stays inside, and that's really what we want. It's like an ivory palace up on a hill that nobody can touch, right? We want to have an intact copy of our DNA that hasn't been bumped into and abused sort of through by meandering through the cell. So it's up there all by itself. However, that makes it difficult, or seemingly would make it difficult to get information out. So what we do is we identify the parts of the DNA that that we need to access, and we make a copy of it so that the original can stay inside the nucleus and the copy can leave. And the way that we make a copy of DNA is by producing an RNA. When we're coding for the production of a protein, the RNA that's developed or that's produced is called an mRNA, or messenger RNA, and that's the first step of protein synthesis. And this process of producing the mRNA is what we call transcription. Once we produce the mRNA, it is able to leave the nucleus, go into the cytoplasm, and act as a pattern for the second part of this process, which is called transcription, which we will see in part 2 of this PowerPoint. That part of that message, as it's being read by the organelle called the ribosome, again, is what we call translation. So we're going to see the whole process unfolding. A little info there about proteins. We're already pretty familiar with proteins, though, so that's not new news for you. All right, so it is important to understand, and we did touch on this idea when we were talking about replication, that we can utilize parts of the DNA, not all of the DNA at one time, which would cause some problem in terms of room available. It makes sense, though, that not all genes are going to be transcribed all the time. We don't need to make digestive enzymes if you're not if you're not introducing food into your stomach and intestines. We don't need to make insulin if we don't have high sugar levels in the blood that we need to control. all kinds of reactions that are occurring in our body only occur at specific times. So one of the roles of DNA is to control what's going on. So the nucleus, the cells receive feedback about what's going on and what needs to be produced. So only portions of the DNA molecule are being accessed at any given time. And we're going to talk about gene control after we finish talking about protein synthesis. It's quite a complex idea, and we're going to look at a simplified model of that. But it is a very complex and a very ongoing study. All right, we can divide the process of transcription and then later translation into three major sort of, three sort of, again, pathways, or three steps. That's a better word. 3 steps. So it's kind of like the beginning, middle, end idea. So initiation is what we call the beginning part. And again, what are we initiating here? We're in the nucleus. We're going to open DNA, and we're going to make an RNA copy. So to begin this process, we are going to introduce an enzyme, and because we're building an RNA molecule, the name of this enzyme should not be a surprise: RNA polymerase. The RNA polymerase is going to bind to the DNA molecule, and when I say upstream of the gene, that's a legitimate sort of biotech or a DNA type word. Upstream of the gene means that it's going to attach before the beginning of the gene, just before the beginning of the gene. Sometimes it's quite a way up the DNA, if you will, at a region that we call the promoter. And I did mention before that promoter regions, these places where we have initiation, are going to have high numbers of A's and T's, which makes them a little bit easier to break, rather than lots of C's and G's with their 3 hydrogen bonds. So we've got this initiation with the RNA polymerase binding to the DNA molecule. Upon binding, the DNA strand is unwound, and it's going to expose the 2 sides of the DNA. The RNA is only going to be built on one side, so we're going to be base pairing on one side, and that side of that strand of the DNA we are going to call an antisense strand. So that strand is going to be used as a template, and the RNA that's going to be built is going to be base paired and therefore identical to the DNA on the other side, which we'll call the sense strand, except for the U's. So I sort of like this diagram. If you look at sense and antisense, or the template and sense strand, they have labeled here the top 2 strands, that would be your DNA. And I think it's a little bit misleading because they have the messenger RNA, it looks like, also attached to that template strand at the same time, which of course wouldn't happen. The DNA would open up and those nucleotides would be added into one side, sort of like we saw in replication with DNA polymerase. So it's a little bit misleading unless you know what you're looking at here. Again, the top 2 strands are DNA, and the bottom strand here is just showing that that the messenger RNA is going to be built on the template strand. And if you compare the very top strand, the sense strand, and the very bottom strand, the messenger RNA, you can see that they are indeed identical, except for the U's being substituted for the T's. Please note, when this happens, it'll be building in the 5' to 3' direction, just like we build new DNA. So the middle part of this step-by-step process is called elongation. And really what's happening here is that RNA polymerase is adding new nucleotides, and we can see that in the diagram here. We can see that we've got a 3' end of an A to which another A is being added. They are RNA nucleotides, so they have the ribose sugar, not deoxyribose. And they're being added in using the big enzyme, RNA polymerase, which is sort of drawn as a big bubble around the whole area here. And that A is going to be introduced, building that strand in orange from 5' to 3'. This will carry on until we hit the end of the particular piece of DNA that's being transcribed. So then we hit the last phase, which is termination. The RNA polymerase is reading along, putting in the nucleotides, and then it reaches a terminator sequence. And that terminator sequence is going to cause the RNA polymerase, or the RNA itself, to detach, and the RNA polymerase to remove itself from the DNA template. And that RNA polymerase can go ahead and find another promoter region, build a different RNA, or the same one again. Sometimes we'll have multiple, or often, usually, we will have multiple copies of the same RNA because we need to make lots of that particular protein in our cell ribosomes. So RNA polymerase may go to transcribe that same gene again, or another one. Okay, and again, this is that particular idea. The DNA is winding up behind it. The RNA polymerase senses the termination sequence, and the RNA is going to detach. So now we've made our RNA, but it's not a mature mRNA. It needs some work on it before it can leave the nucleus. So we have what we call post-translational. No, that should be wrong. Please change that. That is post-transcriptional modifications. That is a mistake. Please, in your mind, change that. It's post-transcriptional modification. So after transcription, still in the nucleus, and we need to make some modifications. So one of the things that needs to happen is a 5' cap. So on the 5' end of the molecule, we need to add a cap, and it is a modified guanine molecule, and it's going to help with initiation of translation once the mRNA is sent from the nucleus and associates with the ribosome. The 5' cap basically tells the ribosome sort of where to begin and how to set everything up, because not only did we build the mRNA from 5' to 3', but we're also going to read it 5' to 3'. It also helps a little bit with protection. On the other end, we are going to put what we call a poly A tail, and it's a very long set of adenine base pairs at the end. They're non-coding, and it has, the poly A tail seems to have an effect on ability to translate, and also on the stability of the molecule itself. I think you'll find, if you have a look, that we're still trying to work out exactly the role that poly A tail plays. And in fact, it's been researched in relation to aging. And the idea of that came up a little bit with Dolly the sheep. The poly A tails in older cells are shorter than the poly A tails that you will find in younger cells. So there seems to be some relationship there. Okay, and there's a visual of what that might look like. We've got the cap on the 5' end, we've got the poly A's on the 3' end, and then the reading frame in the middle, or the coding mRNA. All right, one last thing that we need to do. Evolutionarily speaking, we have a lot of DNA that may not actually code for the particular protein that we're trying to manufacture. So what will also happen is splicing. And there's a special enzyme or complex made of proteins, so like enzyme type proteins, and some RNA thrown in there too, that will cut pieces of this mRNA out and fuse the rest of the pieces together. So there are two, we name them in two ways, exons and introns. So at the very top of this graphic, you're seeing the immature, you're seeing the DNA, then you're seeing the immature mRNA, which is identical to the original DNA. And then you're seeing that the introns, the blue pieces, are being cut out, and the exons are being fused together in the mature mRNA. So those blue parts, the introns, stay in the nucleus. They're not coding, they don't leave, they stay and they can be reused. And the exons are going to exit the nucleus as part of the mature mRNA.